This is the internal structure of the ExiPrep 9.6 Lite. It consists of the magnetic rods cover installation area, Plate 1, which holds buffer cartridge 1 and 2, Plate 2, which holds buffer cartridge 3 and 4, Plate 3, which holds buffer cartridge 5 and 6, and an elution block where cartridge 7 containing the extracted samples is installed. The ExiPrep 9.6 viral DNA RNA kit includes 7 types of buffer cartridge, magnetic rods cover, sealing film, proteinase K, and a user manual. One cartridge set can extract nucleic acids from 96 samples, and each kit contains a total of 4 cartridge sets. Open the door of the ExiPrep 9.6 Lite device. Perform the operation initialization before turning on the device. Manually push plates 1, 2, and 3 inside the device, and turn on the main power switch at the back of the device. After confirming that the LED on the front panel lights up blue, press and hold the power button on the front for 3 seconds to turn on the device. Confirm that the LED lights turn yellow. The device will start the self-test. When the self-test is complete, press the X button to return to the home screen. Once the nucleic acid extraction device is ready, prepare the nucleic acid extraction reagents. Check the kit box for the do not reuse, mark, storage conditions, and expiration date. Open the kit box and inspect the components for any damage. Check each buffer cartridge well's walls and tops for any remaining reagent. Especially, ensure that the black bead in buffer cartridge 2 is at the bottom. After checking the cartridge, move all buffer cartridge to the clean bench. Remove the white tape from the buffer cartridge and open the lid. Carefully peel off the foil sealing film. Be careful to prevent any solution from splashing. Close the lid again. Prepare the remaining cartridges in the same way. Add 1,250 microliter of nuclease-free water or DPC-treated distilled water to proteinase K. Dissolve by vortexing or pipetting. Prepare buffer cartridge number 1. Dispense 20 microliter of a dissolved proteinase into each well. Dispense the samples to be tested into each well of buffer cartridge number 1. Close the lid to complete the preparation of buffer cartridge. On the home screen, touch the plate top button. Install the magnetic rods cover by pushing it all the way until you hear a click. Install buffer cartridge number 2 first, then number 1. Align and install it correctly with the corner parts. After installation, make sure to remove the lid. On the home screen, touch the plate top button. Install buffer cartridge number 4 first, then number 3. After installation, make sure to remove the lid. On the home screen, touch the plate top button. Install buffer cartridge number 6 first, then number 5. After installation, make sure to remove the lid. On the home screen, touch the plate top button. Finally, install buffer cartridge number 7 and remove the lid. Close the door of the ExiPrep 9.6 Lite device. On the home screen, touch the Assign Sample tab. Touch the Auto Assign button to automatically input the sample IDs. Select the checkboxes to apply. On the home screen, touch the Standard Protocol tab. Select the protocol for the kit you will use. Check the selected protocol on the home screen. Check the device operating time under Remaining Time on the home screen. Touch the Time section to switch between running time and finish time. press the Run button to start the extraction process. Check the checklist window and proceed with the experiment. Wait while nucleic acid extraction is in progress. You can check the progress on the home screen. When extraction is complete, open the device door upon notification. Cover the extracted samples with the provided sealing film and transport them to an appropriate location. On the home screen, touch the plate top button. Remove the magnetic rod's cover. Close the lid and remove buffer cartridge 1 and 2. On the home screen, touch the plate top button. Close the lid and remove buffer cartridge 3 and 4. On the home screen, touch the plate top button. Close the lid and remove buffer cartridge 5 and 6. After the experiment, wipe the inside of the device with 70% ethanol on a paper towel. Close the device door. On the home screen, touch the UV lamp and select 15 minutes to start sterilization. The UV sterilization will automatically end after 15 minutes. Press and hold the power button for more than 3 seconds to put the device into standby mode. Turn off the device by switching the power off at the back of the device.